Okay, welcome back everyone. I hope you got recharged by some good coffee and food. And I'm Francesca Finotello from the University of Innsbruck, and I will chair this session focused on single-cell dynamics. Our first speaker is Laleh Haghverdi from NDC Berlin, who will speak about the dynamics of cell differentiation. Please.
Thank you very much. And thanks for the organizers for the nice meeting and the invitation. I enjoyed it very much. So what I am going to talk about is...
Do you have the microphone in? Yeah, maybe pick it up.
Okay. Yeah. Is better? Okay. Is basically how we want to analyze cell differentiation data, and here I am showing a typical example of how this data is looking. So this is an example from the science paper on zebrafish embryo development, that you have multiple time points of snapshot, single-cell snapshot data, and then you want to learn like the differentiation trajectories from one initial state, how you can predict where the next states in future in time will be, and these things. And then, for example, here you get this very complex manifold embedding of this very complex data put up together. And there is basically different things that you need to, like, if you to fully be able to answer this question. So maybe you need the dimension reductions, you need to integrate data from multiple points, and then other methods to improve, like trajectory inference, and then exactly which way cells can go this way or that way, they have plasticity or not. So, and this whole thing started from my PhD time when I was working with Fabian, Florian, and also Maren. So I show some of the publications from that time. Nice to be here again together, all of us. And then, yeah, these are the different parts of the questions that I have already done some. So these are interesting for me, and there are some contributions to these multiple parts, and you can, of course, find other parts. Yeah, so we had this batch correction paper using mutual nearest neighbors. I mean, it is not only batch correction, but data integration can be between different time points. And then more recently, there is a publication from my own group now, which is dealing with the RNA velocity that I'm going to talk about as well. But let's start with the very beginning of the problem. Like when I was doing my PhD, we started to think of cell differentiation as a diffusion-like process. So we say that cells, I mean, there should be some continuity in paths. Okay, you cannot jump from here to somewhere completely different. Of course, it changes. I mean, if you consider one transcription factor is activated, it calls many other genes. So it can look like you go to separate places in your phase space, but fundamentally it is a continuous process that happens on the molecular level, and cells change their state little by little. So there is some stochasticity in it. There is some directional term, like the forces that push the cells towards more differentiated states, and then you can have cell death or cell birth. So all of this you can put in a dynamical equation. So this is called the diffusion drift, or sometimes the Fokker-Planck equation that you see, like that describes how the probability density here shown by P of a time at a specific, at your phase space changes over time. So the derivative with respect to time consists of these three different terms that I just explained. And then from single-cell data, of course, we don't have, it is a bit difficult to get like the drift and birth and cell death and birth rates at different positions of your phase space, but still the geometrical part, which is the random diffusion part, can generate the same manifold. And that's what we actually used in diffusion maps. So in the next slide, I'm talking about it. But first, something that I need to point out is that this Fokker-Planck equation is the continuous space representation, but basically you can pull everything in discrete space into a matrix form. The density probability at one time step can be explained by the previous time step, an operator, a matrix operator acting on it, here shown by pi. Okay, so that's how we came up with diffusion maps. I mean, diffusion map existed, but this intrinsic link to the cell differentiation dynamic was our main first motivation. There were also other components, like diffusion maps are particularly robust to noise. They keep the continuity of the data, unlike TSNE or UMAP, which can break your data into small pieces. And then, for example, we showed that it works better on some data set here, mouse embryonic development, like you see with the diffusion map, the more nice trajectories. And then we developed on it with diffusion pseudotime. And again, using the geometrical part of the information, you can separate the branches and get the pseudotime from it. Now, I want to connect all of these things, like this diffusion drift understanding of the problem, with the more recent methods that have been introduced in the field and used for single-cell data analysis, and that's optimal transportation. Optimal transport, and then the idea is that you have, again, different time points of your data, and then you ask what is the easiest way of moving this distribution to the next time point. And so this I am borrowing from the Cell paper 2019. So yesterday we also heard a talk from Marcus about optimal transport and its application. But yeah, basically, so the optimal transport is finding the transition matrix between one time point and the next time point, ti to ti plus 1, and then it minimizes the cost, so which cells give rise to the cells in the next time point. And here again, there is you see 3 terms. One is the cost function, so I will show in the next slide that how it relates to the drift. There is, and these 2 terms are basically birth and death, cell birth and death relations. So, I mean, very thoroughly speaking, the diffusion drift gives you an explanation of the transition matrix between different cell states in both of your time points. And then in optimal transport, what you try to get is the transportation from the cells in time point 1 to the next time point. And basically, the optimal transport is kind of integration of the diffusion drift equation. And basically, like if I have 2 states, P1 and P2, my transition matrix to the power of T will end up somewhere that I care only about the transitions between cells in the first time point to the next time point. Yes, so again, as I told you, so in the diffusion drift explanation, you can write the transition matrix as some of the 3 components, like diffusion drift and the birth and death rate. So birth and death is basically how you normalize your data, right? So if the if you sum the probabilities to 1, it means that you have no birth and death. If it is bigger than 1, it means that this cell can proliferate. If it is slower than 1, there's some probability of dying, right? So, And then you, as I said, look at it at multiple time points, and then you ask, what is the P that, the pi matrix that brings P1 to P2? And then the expectation of observing like P2 and P1 is just, you can basically state it as a expectation log likelihood maximization. And then, yeah, you can see like there is this immediate resemblance. I must say I didn't get exactly this thing, but when I calculated it, maximizing log likelihood, I get something very similar, and you can already, I mean, something maybe I did wrong, but anyway, so you see these different drift, diffusion, and cell birth and death rates also in the optimal transport formulation. So, yeah, I mean, so far so good, but also like, as was discussed in other talks, there is some relation with the velocity as well, right? So cell state velocities like RNA velocities. And that's when you connect the two terms, like the geometrical and the drift part. And then you can say the derivative with respect of time of P is this, the divergence of the flux. And the flux, like how cells go through, right? So it depends on velocity. So it is, the flux is how many cells go through and with what velocity, right? So, and this is how everything like connects together. This is where RNA velocity, or I mean cell state velocities, it doesn't have to be inferred only from mRNA, but basically the velocities, cell state velocities that you measure is the addition of some stochastic diffusion part and some deterministic that pushes your cells in a specific way. And then if you have these velocities, then you can do things like, for example, cell rank is doing, like with Markov chain, connect many different velocities together to say, if I start with this initial state, what is the probability of ending up here or there? Okay, so yeah, so that's why RNA velocity was also important to us as part of completing this answer for this bigger question. And so just a very quick recap of RNA velocity. So if you have two different stages of gene regulation, like for example, mRNA, unspliced mRNA and spliced mRNA, it adds a dimension of time to your data, because the unspliced will become spliced later, so it can kind of predict in which way your cells are going towards. And then there is this publication from Lamano et al. 2018, who introduced this concept, and then you can write the master equations, what is the probability of going from unspliced with some rate to the splice, and then you have some kinetic parameters and try to find these parameters from your data. And this happens gene-wise, and then you fit these parameters specifically for each gene, and then you try to make up the high dimensional velocity vector by summing up the different components from different genes. So there was also another improvement to the original RNA velocity package Velocito, which was SCVelo. So yeah, so where Velocito, the initial model, was only in the phase portraits of unspliced and unspliced RNAs. It was only using these two ends of your data to fit a line, a steady state line, and inferior parameters, and then SCVelo tries to fit a full trajectory. And my students Valerie and Bridget. So did this fantastic work of looking into these two methods and then finding quite a few places where there could be improvements on pre-processing parameter estimation and visualization step. And this is now, our paper has been accepted last week. So yeah, I mean, there's also a bi-archive version, but wait for the true one, it's better. And then they came up with two different workflows that we call Kappa-Vilo and Eco-Vilo. So Kappa-Vilo is more built up on the classical framework that was introduced in the two other previous methods, and Eco-Vilo is just taking a shortcut. So it's a bit different, and it doesn't decompose the velocity components into different genes and does not try to add them up in the later, right? So, and this eliminates a lot of processing steps, parameter estimations, so which can be sources of errors. And then, I mean, I should say none of these methods are still perfect, but I can say that we did some improvements at some of the stages. For example, on the visualization part, we show that I'm just realizing I'm too slow. Okay, so but on the visualization part, we simulated the data where we knew the true dynamics, so we knew the true velocity vectors. We simulated in such a way to have more heterogeneity in the beginning of the trajectories and then more directed and faster velocities in the end. So the PCA projection is the ground truth because it is, we talked about interpretability and it's a linear method, and then what you see in the PCA, as I say, is the ground truth of our simulated data, and then we look at how SCVilo does the projection. I must say the way SCVilo and Velocyto do the projection, it is very similar. So I mean, there is little differences, but basically, so the approaches are more or less the same. So, but we see that SCVilo is killing that heterogeneity of velocities in the beginning, right? And then it aligns everything, and then also the lengths of the vectors are not according to the ground truth. I must say, the velocity heterogeneity is quite an interesting component, right? So because if we only wanted to know the overall direction, probably the geometrical part, like that pseudotan, would do it, right? So what extra bit of knowledge that we want to get is really this particular cell. Is it always going this, or can it go a little back, or can it go into multi? So keeping this heterogeneity would be nice, actually. And then what we propose in the Kappa-Vilo is using the Nystrom projection. So Nystrom projection, we also used it back then for mapping new data onto diffusion maps. So it is a general framework for mapping new data to old data. And here our new data is the end of the velocity vectors that we try to project on the initial state, which is the cell state. And I just wanted to say, the reason, SCVilo seems to be more and Velocyto, both of them. So they, as I said, they smooth a lot, and then they are more interested in the overall direction. So, for example, how they use it, they use the cosine correlation between a vector and the neighboring cells. And as you can see, so this thing kills the vector norm completely, right? So, because cosine is only about the angle between them, right? So you already in the equation. In the paper, if you are interested, you can see more details on it. I must say that there is a limitation with all nonlinear projection methods, including our method. The Nystrom approach, which we applied here. And that's because we can only project new states which are very similar to some of the existing states, right? So out of the distribution, it is not defined in our existing map. And then, yeah, so there is some limitations, but at least so we keep the heterogeneity and vector lengths a bit better. Okay, yeah, there was something about like the parameter inference. As I said, we try to add up different velocity components, and then there is some scale invariance in like the ODEs, the differential equations that I showed you in the first RNA velocity slide. It means that if you, for a gene, you infer alpha, beta, gamma, so kappa times each of these parameters could be also an answer to your equation, right? And then it is very important, like what is the this kappa for each gene, because that's how big each component is, right? And it can directly completely change the vector direction. So how Velocyto was dealing with is assuming beta equal to 1, and SCVelo was assuming a constant time for all genes, like constant cycle time, like upregulation, downregulation takes the same time. And then we thought, okay, maybe we can just make a bit more realistic assumptions. And then take the cell densities, like in different parts, as some notion of how fast they are moving there. So if the velocity is fast, the cells are staying more in the same place, and then you get more higher cell density. And that's how we try to scale like these different kappa for these different genes. And the assumption is here that we assume uniform cell bears and deaths, right? So because if you have higher cell bears at some point, it changes your density as well, right? So, I mean, again, so this is when we try to come back to this diffusion drift equation, that would be a burden because we want to see them as different terms. But still, we think this is a more realistic assumption in many settings where really in your RNA-seq data, you do see that in fast transition places, there is less cell samples. Okay, so yeah, we applied this kappa-Velo on the pancreatic data and compare it to SCVelo. As I said, because there are different levels, you see SCVelo together with the kappa-Velo's visualization and all SCVelo pipeline. And then we could infer some fast and slow genes based on the cell densities. And then we also applied on the hematopoietic stem cells. And there, actually, the trick is to use the known hierarchy of the cell types, because here I am showing some genes that are very tricky to know whether I want to fit this trajectory to the splice and unsplice, right? And for some of the genes that have, especially the ones that are activated, I see the figure is not appearing very clear, but some genes just get activated at the end of the branch. So then it is only upregulation. And then the data is it is very difficult to assign a good trajectory to it, to say whether it is upregulation or downregulation. And that was basically what is making the hematopoietic stem cells system very difficult for RNA velocity. And there were also other issues with it, like the multiple kinetic genes, so there is more discussions in our paper. So, okay, yeah, I quickly also say the idea of eco-velo. Again, it came from this experiment in the original RNA velocity paper, that they saw that unspliced of a particular time point is predictive, is similar to the spliced counts of the next time point in a circadian genes system of mouse, like mouse liver. So this was bulk data, but basically we say, we try to bypass all these parameter estimation things, and then we can write, so from the ODEs, we can write the velocity as these equations, and then we fix the delta t exactly to the time that you need to degrade all your spliced RNA, right? So if I fix delta t to 1 over gamma, so my nu s will be just, so I will get s times 1 minus 1, which is 0, and it will be only coming from the unspliced counts from the previous time. And that's how it allows us to look for the nearest neighbor of the unspliced counts in the spliced space, and then say this is where this cell is going to move. And then we applied again on both the pancreatic data and the hematopoietic data. It is very interesting. I mean, so for the hematopoietic data, we also get reverse velocities, so they are going in the wrong direction, but that is also what was happening with ST-Velo and like a velocyto, right? So, I mean, it's not perfect, but it is almost doing as good as a ST-Velo, although it is much simpler. So, yeah, but on the pancreatic data, it works quite well, and also on our simulation. Okay, so come to the summary and outlook. I must say, like, this RNA velocity, maybe something, here are the packages. So it's that you can, run like EcoVelo or CappaVelo on your data if you are interested, and the notebooks of these examples that I'm showing. I mean, I talked about a few points, how we try to improve the RNA velocity estimation, but really it's something that I didn't talk about is the data quality, right? So, and that's maybe the biggest burden in inferring cell state velocities, and that's because it's very difficult to assign the mRNA reads to spliced or unspliced. And especially if your genes have very short introns, then it gets more tricky. And I mean, overall, the data reliability is too bad, right? So, and then I think one immediate thing that people are thinking about should be improving the data quality. For example, as I said, if you have any 2 stages measurement of gene regulation, it does add a dimension of time to your data. It doesn't have to be mRNA spliced and unspliced. Of course, the time scale is different, right? So, but for example, if you take mRNA with protein, right? So these are also 2 stages with exactly the same dynamics as we write for the spliced and unspliced counts, right? So mRNA turns into protein, and then there is some degradation of this. So it is exactly the same master equations, right? So it is very nice to just apply it on some other 2 types of modalities, and try to improve ways of reading spliced and unspliced counts. Okay, so very short summary. I talked about the models for differentiation, cell differentiation dynamics, and then discussed the 2 existing models like diffusion drift and the optimal transport, and showed that in both of them you have to infer these 3 terms. Then RNA velocity gives you the sum of the 2 first terms. Yeah, reliable measurement, I said, are 2 packages. And yeah, there is still things to be improved. And we have PhD or postdoc positions, if you are interested in any of the topics I talked about, please also get in contact. And I want to thank especially Valerie and Bridget, who derived this project of RNA velocity, and Fergal, Marike, and Yasmin, also the co-authors. The hematopoietic data was generated by the Marike's group, and other members of my lab, and Melanie, sorry, I forgot. And Melanie as well is part of my group, shared PhD student. Thanks for your attention.
Thank you very much for this beautiful talk. Are there questions? Please, Fabi.
Yeah, thanks, Laleh, for sort of continuing to think about the dynamics of the differentiation. I don't know if you saw, we had this 2019 pseudodynamics type of thing. This was where we sort of on the low-dimensional latent space learned a population model also with a PDE type of model. It's related to diffusion drift, but one thing that we learned there was that we could actually, if we really fit a model where we quantified in various time points the absolute number of cells, we could actually make sense out of the birth-death term, which is in most cases ignored. As you said, it's like a normalizing factor. But it's actually exciting if, in some places of the differentiation dynamic, there's new cells popping up. There's sort of a boost of proliferation or so. So I'm wondering if you sort of could maybe think of a modified version where maybe with these also sort of detailed velocity type models we could learn about birth and death.
I mean, in the two formulations of optimal transport and the diffusion drift, you want to infer these three terms, right? So what you see in your data can come from any of these three components, and it is a bit difficult to only from expression data say this says.
Sure, it's a normalizing, so you need to. So we had external measurements of the absolute number of cells. That could help, right? At various time points. So say first 100 cells, then 1,000.
Yes, but I mean, let's you have 100, you have 1,000. So this 1,000, did they come from this point only? Maybe all of the other ones died, and only from where it came from. So that's why measuring cell birth and death rate at specific positions, I think, would be nice.
Got it.
Thanks a lot, Laleh. I feel like I've learned a lot. Very lecture-style overview is really cool. Do you find that when you're making this ergodicity assumption that density tells you something about speed in the individual gene plots, do you find that you have different gene-specific speeds for different cell types? And how does that change how you predict future state?
So different genes have different speeds, right? So as I said, you can have fast genes which go up and down very fast, or the ones that go slowly, they change over the whole your differentiation process. And so in the end, the velocities that you infer is combining these different components from different genes, and then it should be with the right scaling that we use kappa here for, right? But another point is that when you do this, so you are assuming that my velocities are smooth, right? So if I know the velocity of this gene, I can scale it down and add with the velocity of that gene to get the high dimensional thing. But if things are more noisy, then the average velocities, they cannot be scaled up or down, because at each point the velocity is going to be different. Right, so another point that we also discussed in the paper that I didn't show here is exactly this, that we should care about what time scale we are talking about, right? So if you select different genes, you might get select different set of genes, you might get different velocities as you get from another set of genes which are working in a larger time scale. The same way that you, for example, if you infer velocities from protein mRNA or ataxic mRNA. So these are all things that happen in different time scales, right? And they necessarily do not have to be the same, especially because, for example, in longer range, the diffusion part becomes smaller, right? So the drift is more, but if you talk about more smaller time scales, then the diffusion part is bigger, like you have more stochasticity. And so, I mean, if you chain the little velocities together, it should give you the larger scale velocity, I mean, at least theoretically.
Okay, if there are no other questions, I think we can move on with the next talk. Thank you again, Laleh.